Thank you everyone for joining this evening's webinar for the American Society of Geriatric Otolaryngology. I'm Selena Briggs, the current president of the American Society of Geriatric Otolaryngology. This evening, we have the pleasure of having Dr. Pinto present to us on sensory aging, lessons from the olfactory system. Dr. Pinto is professor and director of research in the section for otolaryngology, head and neck surgery of the Department of Surgery at the University of Chicago. He completed his doctorate at Stanford University and residency and fellowship training supported by an NIH T32 award at the University of Chicago, where he later joined as faculty in 2004. Dr. Pinto's clinical practice centers on endoscopic approaches to the upper airway, including inflammatory disorders and anterior skull-based tumors. His NIH and NIA-funded research investigates risk factors for olfactory and airway disorders in multi-faceted programs involving collaboration across multiple disciplines, including epidemiology, gerontology, environmental health, sociology, and genetics. He also serves as the Vice Dean of Faculty Affairs and the Biological Sciences Division. It's my pleasure to introduce to some and present to others Dr. Pinto.
Great. Thank you, Dr. Briggs, for that nice introduction, and thank you for inviting me to speak today. And I'm very happy to share some of my thoughts about sensory aging, specifically in the sense of smell. I don't have a disclosure side. I do receive some fees from Regeneron and Sanofi and OptiNose for work that's not really related to this, what I'm going to talk to you about here today. So I'm going to start with this slide. This is a diptyque candle.
Can you share your slides? We're not able to see your slides.
Oh, sorry. Let me share. Sorry, I must not be sharing. Thank you. 1 second. Okay, can you see that now?
We can see it now.
Great. How about that? Is that good?
That's perfect. Thank you.
Great. Sorry. I keep thinking like I'm never going to have to use Zoom again, so I'm really not going to have to learn all these things, and somehow that has not gone away. So I'm showing you here this diptyque candle, and I was actually amazed to learn that scented candle market is a $4.22 billion a year industry. There are over 10,000 different candle scents you can get. I don't know if some of you buy Yankee Candle. That's just one of them, many different brands, fancy ones, not fancy ones. 75% of candle buyers say it's extremely or very important. The fragrance, people use them quickly. They use them within a week. They burn them all over their homes. And I think this is important because it tells us that the sense of smell is pretty important to people. And this is true in, you can imagine in scents all across the consumer goods that we use every day. So the sense of smell really allows us to interact with the world. I think this is true of the other special senses as well. Detecting odors in the environment and being able to think about what they are and getting pleasure from that is really important. The sense of smell plays a really important role in human physiology. It's important in perceiving pleasure. It mediates a sense of flavor. So most of what we feel when we're eating food that's delicious is really flavor, which is really sense of smell. It allows us to detect danger in the environment, in particular smoke, but other gas leaks and other dangerous noxious odors, so that we avoid breathing them, so that we avoid ingesting them. It's important in memory, very closely tied to memory, and I'm going to get to that in a little bit in the talk. So you can imagine, how good you feel when you imagine the smell of your grandmother's cookies or certain odors from your childhood, really closely connected to memory. And that's partly because the olfactory system is an ancient part of our nervous system, evolutionarily very ancient. You can imagine even a single cell organism needs to have chemoreceptors to move towards nutrients and to move away from toxic chemicals. So really closely related to the core functions of our brain, including memory, and similarly affects and is plugged into emotional mood centers, and affects how we feel. So, and this is, why aromatherapy makes people feel good, because those scents again, give pleasure to people. And these things are all critical parts of our lives. We sort of take them for granted. And as I'll get to, now we have lots of people who have smell loss, where acquired smell loss from viral illness, from COVID. And now we're getting to an era where people who appreciate smell loss understand how much a burden it is. And of course, the most common cause of smell loss is age-related smell loss. And so we have many older people who have poor sense of smell. A huge proportion of people who are older can't detect the odor in natural gas, the warning odor. So this has a lot of implications for health and quality of life of older people. The one thing that's interesting and kind of unique about the sense of smell in terms of the special senses is it's wired directly into our brain. So as you know, you can inhale an odor. Those odor molecules get transported up to olfactory receptors by chaperone proteins in the mucus of your nose. Those odors then bind olfactory receptors, or a suite of olfactory receptors, a suite of odors and a suite of olfactory receptors, creating signaling, and that signal passes through the olfactory tract and doesn't synapse, goes straight into different parts of the brain, including the hippocampus for memory and the orbital frontal cortex involved in emotion, and many other places. So the olfactory system is actually plugged in right into the brain, and that's probably from an evolutionary point of view, and also because this information is quite important to us. So when your sense of smell declines, especially with age, you have a lot of problems. And particularly in the kinds of things that older people do, what we call activities of daily living. So things like detecting spoiled food, smoke, gas leaks, eating or cooking, which we all do every day, personal hygiene. So one thing that comes up for these patients is that, they can't tell if they need a shower. They get some social anxiety about odors because they can't detect them even on their own body. They have trouble with cleaning, have trouble buying fresh food. They can't smell strawberries, right at season, or other foods. And then a lot of what we do in terms of social function is eating. So you can imagine going out is not nearly as pleasurable as it could be in terms of the social aspects of being in a meal together. And so many of these people who have this problem really don't like to go out with other people to dinner. Another reason is that when you have a really poor sense of smell, all the food tastes the same, tastes like cardboard, has no flavor. That leads to reduced weight and appetite. So there's some dietary consequences. You can have decreased psychological well-being. There's increased symptoms of depression, and I'll show you some data on that. And you have an overall decline in pleasure and quality of life. So I'm gonna, with that sort of nuts and bolts of the importance of olfaction, I'm gonna walk you through a series of studies that we've done looking at some different questions on how sensory function is really important for older people. What are the risk factors for sensory loss, in particular the olfactory system? And then what are some of the consequences of olfactory loss for older people? A lot of my work centers on a project that I'm part of called the National Social Life Health and Aging Project. This is a multidisciplinary project involving a number of different investigators. It's led by a sociologist and a survey epidemiologist. The group has studied 3,005 community residing older adults aged 57 to 85 at baseline, starting in 2005. This was a representative probability sample of the United States. And so the results are generalizable to that population. Professional interviewers went into the homes of these people all over the country. They collected a host of detailed information about health and social function with standardized health questionnaires, and then also collected biomeasures, a number of different biomeasures, one of which was objective measure of the sense of smell. There were multiple rounds of data collection. So in 2010, we went back for 5-year follow-up of the original participants who were still alive and then their co-resident partners. In 2015, we went back to all of those people again, and then started a new cohort of baby boomers. And then we've just finished a data collection and are about to start analyzing the results of our most recent wave, which is 15-year follow-up. So we have longitudinal data on the survivors of this cohort. And we have a host of information. We have, demographic information. We have cognitive function. We have self-rated physical health. We have comorbidities in terms of their medical conditions and indices for that. We have mental health measures that are validated, such as depression and anxiety and stress. We have smoking and drinking. We have education and also assets. Both are measures of socioeconomic status. And there are people in this project who have studied everything from elder abuse to medications that conflict, because we have good medication histories, to sexuality, diabetes, many different diseases attacked from many different angles. So it's really a very rich data set to study. It's a public data set. You can download the data from the website housed at the University of Michigan, and really useful to answer questions about aging and health. I should also mention that we do have some measures of other senses, including vision, hearing, taste, and touch. And those are also obviously relevant to our field, relevant in aging, and have some interesting co-relationships. So how do we measure the sense of smell? We used odor-embedded markers called sniff and sticks. We had a set of them, a subset of 5 items, and they were given the odor and then asked to identify it from 4 choices. We then categorized the scores as number correct. So 0 to 1 was anosmia, 2 to 3 was hyposmia, and 4 to 5 was normosmia, so normal sense of smell. So took a median of 3 minutes, very short test. We also measured threshold, and I'll get to that in a little bit, which is at what concentration can you detect an odor? That was a 6-item test. So the 2 tests only took about 3 minutes, and you'll see we can generate a lot of information from those 3 minutes. All right, so I'm going to go through the risk factors for age-related smell loss first. These are things to think about when you're seeing older patients and considering their olfactory function. So the first thing we did was replicate the long-standing finding that sensory function declines with age. So Richard Doty in 1984 published a beautiful diagram of age-related smell loss with the University of Pennsylvania smell identification test, where you can see quite a big detriment of age. And we reproduce those here very nicely. These curves look almost the same. So whether you use a threshold of greater than 3 right or 4 right or 5 right, all 5 right, you see as the people are older, you see decreased proportion of getting the number correct. So age is a really big factor in smell loss. It's interesting that we don't really know the cause of this. So if you do cadaver studies, you can still find olfactory stem cells in older people in the nose. So it's not necessarily a loss of regenerative capacity of the peripheral olfactory system. It could be related to, mitochondrial dysfunction or inflammation over time, or other mechanisms of age-related decline in sensory function. But we really don't have a good answer for that, but it's a very reproducible finding. And it's very useful for, counseling patients, because we know that older patients are likely to have decreased sense of smell. All right. Well, one thing when we did these models, we looked at, we wanted to look at other effects. So we looked at, for example, sex, men versus women, and we found that women had a worse sense of smell compared to men. And this is also, a better sense of smell compared to men. Men had a worse sense of smell. And as you can imagine, that's also something that Doty found and has been reproduced many times. Again, don't have a good mechanism for that, and so we're open to questions about why that might be the case. The other finding that we found that was novel, actually, was that African Americans had a worse sense of smell than their counterparts. And this survived adjustment for a number of different issues and some logistic regression models, which I'm showing you here. So for example, you can see that adjusting for cognition, so better cognition is associated with better olfactory function. More education is associated with better olfactory function. Assets in this model didn't make too much of a difference, but you can see African Americans are more likely to have olfactory loss than their counterparts. And, we thought that might be related to health factors, so we further adjusted for physical and mental health, comorbidity, smoking and drinking. So in this expanded model, you see that odds ratio really hasn't changed. So despite controlling for a number of different relevant health factors and socioeconomic status, mental health, drinking and smoking, African Americans have a higher odds of having olfactory loss. And, we started to think about this, and it'll get to some of the later work that I'm going to tell you about. This could be due to a number of reasons. Remember, these are people that were born between 1927 and 1945, so they certainly could have life course differences. You can imagine how different things were in that era in between these different groups in terms of nutrition, early childhood exposures, health over time. Many things could affect that. Also, there could be environmental differences, so potentially effects of urban environment, air pollution. They obviously have a different genetic ancestry, so there could be ancestry-related susceptibility, cultural differences. So there's a lot of different reasons why this might be the case. And we're still investigating this, and this has been replicated in other populations, other studies. So one thing we sort of thought about directly from that is sort of environmental exposures. You can imagine there's race-based environmental differences in exposure. And we know that the nose is a portal to the brain because the olfactory nerve, in fact, is in the nose and crosses through the cribriform plate and gets into the connects with the olfactory bulb, synapses, and then again progresses onward to other brain centers, as I mentioned earlier. So this provides a potential pathway for not only pathogens, but airborne material, such as particulate matter pollution, to enter the central nervous system. And in fact, if you give radiolabeled particles to rodent animal models, you can see these particles end up in the brain through this pathway. And so the question is, could this affect olfactory aging and indeed in the brain health itself? So we were able to map pollution exposure to our cohort in NSHAP, and we used some spatiotemporal models by a colleague, Helen Su, at Tufts University, and we were able to show that increased pollution exposure, in this case, particulate matter 2.5 microns or smaller, so increased particulate matter airborne pollution was associated with an increased odds of having olfactory dysfunction. So here you can see in model 1, this is adjusted for age, gender, race, ethnicity, and education. And even when you include comorbidity, smoking, and the season of testing, you still see a significant increased odds of olfactory dysfunction in older US adults. So what this is telling us is that at least one reason why older people use their sense of smell could be a lifetime of exposure to airborne toxicants, in this case, particulate matter. And remember, these are people who grew up in an era of leaded gasoline, who grew up in an era before the Environmental Protection Agency was created, before there were filters on factories, all the different things we take for granted now. These people have faced a lifetime of exposure. So this definitely could explain, in part, age-related olfactory loss, and it's certainly a risk factor for olfactory loss in general. I should also mention that, we modeled this exposure, in windows before the smell test was done, in 2005, but, this is going to extend before that. And so, think about that whole lifetime of exposure that could make a difference. And remember, the mean exposure here was, just a little bit above the EPA limit. So we're not talking about exposures of like what people experience in Beijing or in Mexico City or in Sao Paulo or in Delhi, where the levels are, in the hundreds of micrograms per meter cubed. This is at low levels, we can still detect an effect, indicating that environmental influences are important for sensory function in older adults and olfactory function in particular. So that led us to this idea, and our current work is to think about, well, if that's the case, then these particles can injure the olfactory system, certainly in the nose and potentially in the olfactory bulb. Well, then these particles can get into the brain, and they can cause problems with the brain. So you can see here in this cartoon that, these particles can land in the olfactory mucosa. They can be transported potentially into the olfactory bulb, along the olfactory tract, and into these other parts of the brain that are really important, like the basal ganglia and the thalamus and the cortex and the amygdala. All these parts of the brain that I said at the beginning are directly connected to the olfactory system. And that led us to our current project called AirNose, which is the air pollution and Alzheimer's dementia, neuropathologic and olfactory mechanisms in multi-ethnic longitudinal cohorts study. And our team, my co-PI is Jennifer Wave, who is an environmental epidemiologist at Boston University. The people that we study are a dementia cohort at Rush University, led by David Bennett, who's a neurologist there, with his colleagues Lisa Barnes and David Ramirez. We have experts in neuropathology, airborne exposures work from Joel Kaufman and his colleagues at University of Washington. And then we're so lucky to partner with Gunther Oberderstor, who did the animal work and is an expert in toxicology of these particulate matter and how they might get into the brain. And I'll show you some imaging data from our colleague Ushi Graham, who is a nanoimager. So our idea here is that air pollution composed of ultrafine particulate matter can get into the olfactory bulb. There it can cause pathologic disease, leading to olfactory dysfunction and decline, and ultimately ending up in cognitive problems and dementia. And I should mention that olfactory loss is a precursor of dementia. It strongly predicts the onset of dementia subsequently and mild cognitive impairment. This is one environmental mechanism that could connect these two important problems. And so our aims are to characterize long-term exposure to ambient pollutants in these cohorts from the landmark MESA Air project, where Joel has mapped out pollution exposures all across the country. Test the hypothesis that higher long-term air pollution exposure are associated with Alzheimer's dementia and the related outcomes, and then measure, in fact, directly particulate matter in olfactory bulbs using advanced microscopy as a novel biomarker of air pollution. Again, trying to prove this idea that particles can get into your brain through your nose and cause big problems there. So the cohorts that we study, these are all related and linked cohorts for the Memory and Aging Project. Is one of them. So of the religious order study, the minority aging and research study, and the Latino core. So we have a diverse group of cohorts, really diverse population, large numbers. All of these patients get measured every year. We get smell test scores every year, a lot of different health information related to dementia. And then when they die, they agree to donate their bodies to science, where we can get access to the pathology. So they're doing all kinds of work on the brain, and we get the olfactory bulbs where we can look for these particles. So our questions are, what type of particles get into the olfactory bulb? Do they interact with cells? Is there local inflammation going on? Do they undergo bioprocessing? And then our big question is really, do these particles increase neurodegenerative disease, neurodegenerative pathology, and do these nanoparticles translocate to other parts of the brain? And our hypothesis is that people exposed to higher levels of air pollution end up with more ultrafine particles in the olfactory bulb compared to those who live in less exposed areas, and are more likely to develop olfactory loss and Alzheimer's disease. So this is the mechanism that Ushi uses. It's called high-angle annular dark field imaging. We can observe particle location and behavior at a 1 nanometer scale. We can also use energy dispersive spectroscopy and energy electron loss spectroscopy to not only count the particles and look at the pictures, but actually measure what's in those particles. What metal signatures can we find in those particles? And then we can ask how many are there, what are they made of, and how are they interacting with the tissue. So I'll show you some pictures from that. So these are what the particles look like. This is the olfactory bulb at a 10 micrometer meter scale. Here's at a 200 nanometer scale. And you can see there are these carbon particles metal particles with spheres with these carbon coatings around them. And so we do, in fact, find ultrafine particles in the olfactory bulb, which is a little bit scary. And then if we look at with those spectroscopic methods, we can tell what they're made of. And you can see they're made of things like aluminum and bad things like chromium and lead. Again, I alluded to earlier, these are people who lived in an era of leaded gasoline. And so you can see that you can find these toxic particles, the effect of that for particularly worried about lead, which is not a natural thing to have in your body. And so we can measure these chemicals and we can see, what they're made of and what might be toxic to the olfactory system in the brain itself. We can also see evidence of breakdown of these particles affecting cellular structures, subcellular structures like mitochondria. We see some evidence that these nanoparticles can break down mitochondria. So that again could be a metabolic effect of these, which could cause neurodegenerative processes. And again, in this zoom in, you can see some bioprocessing of these particles. So there's some responses where the body sends ferritin as a way of dealing with this, containing this problem of nanoparticles. And you can see these zinc and manganese and chromium and lead and arsenic here in these particles, and sometimes aluminum and silica. So we're trying to get some evidence of what's going on and what effects it's having in the actual cells. And the other interesting thing is these carbon spheres, they have organic material on the surface. So you can imagine all of the combustion-related organic chemicals, and here you see sheets of them on the particle. So it's not just the particle, which is not good, but also what it's carrying, and it's carrying these likely toxic carbon coatings on there of organic chemicals that could be bad for the brain. So, and these are in sort of sheets on the surface there. Okay, so the big question though is that's nice, but how does that correlate with pathology? So we're in the early phases of this work, and I'm going to show you just early data for a handful of these, where we can see some very small numbers, and these are very rough calculations, but you can see here that you have pretty good correlation with the number of particles and important measures of Alzheimer's disease, like the Braak score or amyloid levels or tau density. So pathology of the brain seems to correlate with these exposures, and we'll hopefully have about 400 olfactory bulbs measured in the next few months, and then we'll be able to really dig into these questions. All right, so that's environmental influences, and I alluded to earlier there could be genetic differences, and here's some work we did again in NSHAP looking at genetic susceptibility, and we found that, for example, people who carry the Alzheimer's risk allele of E4, people who have those alleles are more likely to have worse sense of smell. So certainly that correlates. And again, those people are, again, at high risk for dementia. Those two things go together. In fact, you can find Alzheimer's pathology in the olfactory bulb and even in other parts of the olfactory system, consistent with this finding. What's interesting is we found that ApoE carriers had worse sense of smell, and that started at age 75 here. So the carriers are here in black. And you can see in the beginning, there's not too much differences, but at age 75, that starts to sort of trend away. Our numbers in 85 or older are not very great, but if you focus right here, you can see significantly worse olfactory identification starting at age 75 in carriers. But we found that something that was interesting was that if for threshold, again, the threshold of when you can detect an odor, what concentration. So that's kind of less dependent on cognition. You don't have to name the odor. You don't have to think about what it is. You just have to say, is there something there? Yes or no. That tended to manifest earlier, starting at age 65. And so this could tell us that threshold is actually an even better measure of prediction of cognitive loss. And that seems to be occur in carriers as early as age 65. Again, there's some genetic susceptibility here and maybe some gene-environment interactions. We did look at, does that predict change over time? Again, we have olfaction over time. And again, this is in odor identification. And indeed, E4 carriers, people who have the higher risk of dementia, they are more likely to lose their sense of smell faster over time. So they're worse in cross-section, and then they decline faster over time. All right, one thing we're all interested in is what about inflammatory effects? So sinonasal disease is one of the most common causes of olfactory impairment. For clinicians who treat olfactory disorders, this is great because this is the one kind of olfactory disorder we actually have treatments for. It's reversible. And then again, I really personally look at this very closely because this is one thing we can do something about. So asking about inflammatory disease, working up inflammatory disease is really a key step in approaching patients with this problem. 60–80% of people with chronic sinusitis have some form of olfactory dysfunction. It's typically hyposmia, but it can be dysosmia also. It can be anosmia and severe polyps or AERD. The prevalence is really high if you test people, and many people don't recognize that loss. And so consequently, things that increase inflammation in the nose are highly associated with olfactory impairment as it relates to CRS. And these are things like smoking, nasal polyposis, asthma, tissue eosinophilia. These are all markers of increased inflammation and worsen the sense of smell in this context. And so, thinking about olfaction in the context of inflammation is really important because, again, we can fix these problems with surgery, with medicines, with treatments. Definitely something to look at. And, how does that happen? Well, a lot of groups have looked at this very nicely, and you can see eosinophils or ECP as a marker of inflammation associated with olfactory loss, particular cytokines, potentially certain inflammatory pathways that can affect neural function or apoptosis or cell death. And then also you can see structural effects. So you see some decrease in size and function of the olfactory bulb in patients who have chronic inflammation in their nose. Now it's, is that a cause or an effect? It's not clear, but certainly chronic sinusitis can affect the sense of smell through these pathways. All right, finally for risk factors, so viruses, right? Viral smell loss is a big risk factor for smell loss. We've learned about this the hard way with SARS coronavirus 2, and which is particularly olfactory toxic. This has obviously happened all over the world. And so this is, not something that's unique to the United States, really been widely reported where people had large percent, particularly with the earliest form of the virus that's waned over time, but that's resulted in millions and millions of people with long-lasting anosmia. Even some early studies in COVID showed that, some up to 7% of people may have anosmia, up to a year later. Of course, that causes other health consequences, which I'm going to get to, but not the least of which is nutrition and quality of life, mental health. And we're really, going to see whether acquired smell loss, such as olfactory related loss from SARS coronavirus 2 is a contributor to neurodegenerative disease. So I've shown you that and discussed that age-related loss is really closely tied to dementia. And the question is, okay, what about other forms of smell loss? Very difficult to study this, just when you think you can study something, the virus mutates, things change, people don't want to participate. But we're going to have to get creative about figuring out this. And, we have colleagues like Brad Goldstein and Eric Holbrook and others who are working on this problem, doing some really nice work on support cells as being risk factors. But this is something we're going to all see the consequences of. You can think about it sort of accelerated aging. We have younger people who are going to lose their sense of smell early and then face a long time of consequences. All right, I'm going to switch gears now and talk to consequences of smell loss. So mental health is really a big problem after smell loss. It's always been this question about, okay, does the smell loss cause the mental health problems, or could the mental health problems be related to the smell loss, right? Because the brain pathways overlap, and could be different mechanisms, and so that was a sort of open question in the field. And so because we had nice longitudinal data in NSHAP, we were able to test that question and look at people with smell loss. Are they more likely to develop depression 5 or 10 years later? And here in this logistic regression, adjusting for many different things, we show indeed they are. And then we were able to look at the opposite, which is people with depression, are they at baseline, are they more likely to get olfactory loss later? And that wasn't the case, indicating that it does look like people with smell loss are then because of that, getting more depressive symptoms and more interest in depression. Interestingly, that signal was really seen in our older adult population who had no comorbidities, and it was not seen in patients who had one comorbidity, indicating that once you get sicker, lots of things can cause depression rather than just olfaction. But this quite clearly connected olfactory loss with subsequent odds of getting depression. What about other brain health outcomes? So as I mentioned in the Rush Alzheimer's disease cohorts, in the Memory and Aging Project, this project, they also collected MRIs over time, among other interesting covariates. So we were able to ask the question, if you test people for olfactory function and look at their olfactory trajectory, do people with olfactory decline actually end up with changes in their brain themselves, both in cognition and in structural aspects of the brain? And so we had 3T MRI data. We parsed that data into different parts of the brain: the primary olfactory cortex, the secondary olfactory cortex, temporal cortex, frontal parietal region, visual, and sensory motor, to look at, in these patients, if we look at how they decline over time, then what do their brains look like after that, and which parts of the brain are affected? And so this is what the sort of study design is. We have these number of different olfactory tests. Then later they get an MRI, and then eventually they die and they get a final diagnosis of Alzheimer's or not. And you can see here that people who had a faster olfactory decline, as many other people have shown, have worse global cognition, worse memory, worse perceptual speed, and then even have some trend towards other problems in other parts of their brain, other functions of the brain. Again, this is adjusted data for genotype and sex and education and other things. And in fact, that rate of decline does predict the odds of incident mild cognitive impairment and dementia. You can see here just as strongly as that high risk factor ApoE. So, really big risk factor, really big predictor of mild cognitive impairment or dementia. So in fact, studying people, over time really gives you a lot of information about their risk for dementia. And then finally, we saw that olfactory loss was associated with a difference in the primary olfactory regions. So you can see here, those with severe decline, there's a trend here in this line with worse sense of smell decline, with decreased gray matter volume in the olfactory regions. This was also true in the secondary olfactory regions, again, smaller gray matter volumes. When we looked at diagnosis, at the end, those with mild cognitive impairment and Alzheimer's disease, again, show smaller parts of the olfactory, smaller regions of the olfactory system in the primary olfactory cortex compared to people with normal cognition. And so this tells us that cognitively normal older adults who have faster, I should mention this is in cognitively normal adults at baseline, they have faster rates of olfactory dysfunction, and they are more likely to develop impaired cognition and dementia. They have structural changes in their brain in the olfactory regions. And so testing people's odor identification over time can really help us identify people at high risk for Alzheimer's disease. All right, I'm going to finish up here by talking about other aspects of olfactory physiology that affect other parts of the body. So chemoreceptors now, it turns out, are expressed in many parts of the body, including the upper airway and the testes, in the kidneys, in the gut. The question is, what are they doing there? And we started with a sort of a related part of our physiology to the upper airway, which is the lower airway. So olfactory receptors are expressed in the lung. And you can imagine that, detecting chemicals in the air is just as important in the upper airway as it is in the lower airway. Again, probably related to irritants or toxins, et cetera. And so in this cohort, this is work I did with Hong-Lei Chen, who is an epidemiologist at Michigan State. Actually, he's the PI of this project, and he did this work in this cohort called the Health ABC Project. And this is aging and body composition. It's originally a study of nutrition. They had annual visits, semi-annual quarterly telephone visits, and then they measured the sense of smell in 1999 to 2000 with the 12-item UPSID test. Again, odor identification. You can score it good, medium, or bad by the number correct. And then we looked at an outcome. They looked at an outcome which is total or first ever hospitalizations due to pneumonia. If these olfactory receptors are expressed in the lung, they might be doing something, and maybe this is where you could see it. And so we had about 2,500 participants. These were present in a couple of different areas of the country. Memphis and Pittsburgh were the sites. There was, about a 30% history of patients getting pneumonia in the past. There was 12 years of follow-up. And over those 12 years of follow-up, there were 801 hospitalizations due to pneumonia. Majority were the first time. And interestingly enough, those with poor olfaction had a much higher rate of getting hospitalized for pneumonia over time compared to those with moderate olfaction here in red or good olfaction here in blue, indicating that poor olfaction was a risk factor indeed for getting admitted to the hospital for pneumonia. Here's another way of looking at it. Those with good olfaction, had a 25 per 1,000 person years kind of risk of pneumonia versus those who had poor olfaction, 44.4. So big difference in risk for hospitalization. As you can imagine, for older adults, that's a big deal being hospitalized for pneumonia. And so, that translates to quite increased risk of that. And it's interesting, these results were similar across sex or race, and they were adjusted for all kinds of things related to hospitalization for pneumonia. So age, race, sex, location of where they were recruited, education, smoking and drinking, their physical function, brisk walking, body mass index, general health status, and a number of comorbidities. So really kind of a robust relationship. Hong-Lei has suggested, is this an indicator of loss of barrier function in the airway? Is this decreased sensor function and triggering responses to pathogens or airway toxins? Is it a signal of poor airway sensitivity leading to aspiration? And is this just deterioration of chemosensation in the lower airway? So the mechanisms could be any of these things, but this has some clinical utility because you can predict that. Finally, the ultimate clinical outcome is mortality. So this is some work we did a number of years ago. Wow, it's now 10 years ago, where we looked at, does odor identification predict mortality? And indeed, in NSHAP, every increase in error you see a great increase in probability of dying 5 years later. With people who have anosmia, 0 out of 5 score, they have a close to 40%, a little over 30% chance of dying in the next 5 years. You might say, oh, well, that's just due to because older people, there's just more older people, and older people are likely to die. But you can see this is true in the youngest age group, the middle age group, and the lower age group. In each, you see this dose response of those who have anosmia have increased probability of death percentage dying compared to hyposmia or normosmia. And that's true for all 3 age ranges. So, and this has now been replicated in other studies. So really quite a robust predictor of death. We've gone on to do some subsequent work on this topic, looking at trajectories, the trajectory decline over time, an even better predictor. And in fact, it is. So you can see here compared to, so people who change from from who have persistent anosmia over time or have anosmia over time are much more likely to die than those who have a shorter change in their sense of smell. So in conclusion, I'd like to just say that there are inherent and external causes of decline in the sense of smell in older adults. These can be things like age, sex, and race. It can be genetic variation, and it can be environmental exposures such as air pollution. Decline in sense of smell can signal future decline in physical and mental health, especially related to brain function and brain structure, as I've showed you, but also in other parts of our physiology. I didn't have time to show you data on frailty. It predicts frailty, and other parts of our physiology for older adults. We can exploit this, though, to identify people who are at high risk for early intervention, and enroll those people in pre-disease clinical trials. Particularly for Alzheimer's, that's a really great option because you can find people who are at high risk. And particularly since we're in the era of like trying to move earlier with treatments and prevent the onset of dementia, we really want to identify people really early. Even if you can't treat people, it's still worthwhile because you can tell the family, you can tell the patient, they can prepare. And so I still think that's really important. And then looking at change in sense of smell over time, I think is important. We can, those trajectories can really be important, and we can look ahead for those problems that are going to happen in the future. Changes in olfaction predict major health outcomes. They have an enormous impact on quality of life. And then, for this group as otolaryngologists, I think you can think about these concepts in the setting of other senses that are really important to us, in particular hearing. So hearing is also closely related to dementia. We actually have an intervention for that, which is hearing aids or cochlear implants. And Franklin has done some amazing work showing that that can actually potentially stave off and stop the decline in the brain and cognitive function. So we have to think about, the senses that we're experts in more carefully. Of course, there are environmental determinants of things like hearing. There are similar relationships with hearing and other health outcomes. So I think this is sort of a model for sensory aging more generally, and we should really jump into that as people who are experts in sensory function and in sensory aging. So I'll just conclude by thanking all the people who have helped me with this work. I showed you the work that was led by Honglei Chen in the Health ABC study, my partner, Jennifer Wave for pollution. NSHAP pollution exposure was done by Helen Su at Tufts, and then all the work with David Bennett on the Rush cohorts. I have a lot of colleagues here in our olfactory research group here at the University of Chicago, including Martha McClintock in psychology, some colleagues in public health science and geriatrics. And then I showed you work from a number of different medical students, including Lucy Yu, who's at Harvard right now as a otolaryngology resident. And so I will stop there and see if there's any time for questions.
Great. Thank you so much for an awesome and phenomenal presentation. It's interesting, the last point that you had raised a question for me. Actually, I was thinking about it as you were presenting. Are there any effective interventions for individuals with anosmia or hyperosmia that are effective?
Sure. For inflammatory related loss, we have a lot of different tools in our armamentarium. So biologics, topical steroids and other things that can control inflammation. That's our best case scenario. For other forms of smell loss, we don't have great treatments. The one thing we have is smell training, which in some situations has been shown to accelerate recovery of olfactory function, most notably in, and this is pre-COVID studies, in viral-related smell loss. And that's something that, in some of those studies, and they're not perfect, has shown to improve smell function in about 30% of people who do it. So it has no risks, maybe as sort of physical therapy for the olfactory system, stimulates those olfactory neurons or reconnects the damaged olfactory periphery to the brain, and that can be helpful. There is a lot of work in this area. There are companies working on drugs for olfactory regeneration. So I think this is going to be an area to watch. We also have things like PRP injections in the olfactory mucosa to help with viral damage. So there are some newer treatments that show some promise. And we're hopeful that if we can, that would really, test Koch's postulates. If we can fix olfaction and then help stave off those sort of brain sequelae, that would be really a great thing to shoot for in the future.
Yeah, exactly. Seeing if they're having an impact on the dementia as well. What about the olfactory implants? Any comments on some of
Yeah,
that work?
that's great. So, yeah, the group in Virginia is doing some really exciting work on, we have cochlear implants, which we can use to deliver auditory information. And there's certainly the possibility, and they're showing some test possibility of that, of olfactory implants plugging in, for example, into the olfactory bulb and then delivering information. One thing that we're a little bit behind on in olfaction is understanding the coding of how are those shapes of those different odors coded into signals that the brain can understand. But I think that work could have the promise of translating chemical information into signals that the brain could reinterpret and potentially serve as sort of a bionic nose, which would be another wonderful way of doing that. I mean, we'd have to think about how would you miniaturize that and how would you get the electrodes in, and there's a lot of work to be done for that. But I think it's got great promise, and I'm excited about that kind of work.
Yeah, thank you. Any other questions from you?
Jay, that was really terrific. Mike Johns here. I'm from LA. I really enjoyed that talk. The content is exceptional and really well delivered and important. I'm going to send my parents the recording and have them watch. I just wanted to say kudos, and I do enjoy the olfactory retraining. I got one for my kid when he had COVID, and I just enjoy doing it. I think my smell's okay.
Yeah, it's something that's fun for people to do. I'm not sure, why can you not see me here? There we go. Um, it's something that's fun to do. It's now how much it affects natural recovery. Does it accelerate natural recovery or not? That's something that's like a question. But I think it has some promise and it has no risks, right? So it's no risks. And Mike,
Yeah.
it's nice to see you after many, many years at the AGS and in Seattle and some other places. So great to see you doing well in LA.
And yeah, thank you. And thanks so much for doing this for this great organization.
Any other
Hi,
questions?
Jay. Kurosh Farm at University of Connecticut. Thank you very much for an exciting talk for somebody who's had an opportunity to kind of follow your career over the last 20 years. I'm so excited to see what you've accomplished, and it was so enjoyable to listen to your work. And I was fascinated with the multiple dimensions of the work that you presented. And the latest dimension, the impact of pollution, was earth-shattering to me. And I'm wondering what is going up into my brain at this moment. But a couple of questions regarding that. Is there data to suggest people who live in more polluted areas are at greater risk in terms of suffering cognitive decline and dementia that would align with some of the data that you demonstrated? For example, the correlations between your findings and, say, amyloid in the brain and so forth. That kind of data is also supportive.
That's a great question. I don't know the answer to that, but I will answer that in this way. And it's great to see you also at a mecca of smell research center, of course. And nice to see you again after many years. We did look at regionality, and we did adjust for region and still see the same effects, but we saw in the US, there's big regionality to pollution exposure. So obviously, if you're lucky enough to live in the West, where there's less pollution, you have much lower levels. The Northeast corridor has very much higher levels than other parts of the country related to the density and highways and older part of the country. And so their exposures are higher. I don't know if you look at a map of dementia exposure incidence, does that overlap? But I'll go back and look at that. That's a really great question. I think the thing that's scary for me is that these levels we're talking about, the relationships I showed you, they're at just over the EPA limit. The EPA limit's like 12, and these are like mean of like 13 or 14. And so that tells us that we can detect these signals even at pretty low levels. I think, those of us who are here who are a little younger, we've lived in an era where hopefully the pollution levels are less cumulatively over lifetime. But I think this data is telling us that even low levels of particulate matter pollution are not good for your nose. They're probably not good for your lung or your brain or any other part of your body. And, some of the nice studies from six city studies and others show like, the level goes up today, people die of heart attacks tomorrow, people are in the emergency room for asthma, they're quite close correlations. So it's something we need to all be worried about from a health point of view.
Yeah, if I may ask a follow-up question on that same topic, you mentioned the cumulative effect, potential cumulative effects and stuff. And if there was a cumulative effect, wouldn't you expect to find much higher levels as a result of a lifetime inhalation of all these pollutants and stuff, and on your very high resolution microscopy and various tools that you find? Like, once you find that whole olfactory bulb completely covered and penetrated with all sorts of different pollutants, if there was no clearance mechanism by nature to sort of reduce the exposure. So obviously it penetrates and it is then dispersed to other potential areas of the brain. But might there be a clearance mechanism that kind of reduces that risk of spread within the brain tissue?
Yeah, well, I think you should never discount the body's ability to sustain assault. So I agree with, so first of all, I think in the Alzheimer's world, there is this idea of clearance of toxins, clearance of cellular debris, clearance of inflammation is the lack of that is one reason why you get neurodegenerative disease. And so people are really thinking about that pretty carefully. I think we do see evidence of the body trying to engulf these particles, trying to send ferritin as sort of an inflammatory marker. And so we think that there are clearance mechanisms. We do see higher levels in the olfactory bulb compared to, say, the cerebellum that's not connected to the olfactory system. So we do think that it disperses in different places and is differentially deposited. And I think we are looking at other ways of measuring cumulative exposure. So we're gonna start getting lung samples from these patients in a related study, and we're gonna look at like how many of these particles are in the lung to tell like how many terminally deposit there, and use that as a marker to see, does that correlate with what's in the brain or is that less? I'm hopeful that if we could figure out the mechanisms, then maybe we could design, drugs to block those toxic mechanisms, enhanced clearance, or drugs that allow us to process these metals better. And that might offer, a way of solving this problem after the horse has left the barn kind of thing. And that might be
Fascinating
a
work.
therapeutic way to do it.
Yeah, very excited for all your work. Thank you.
It's interesting. Two populations that you might target that you would have higher exposures than normal would be military and firefighters, to see if there's a way of intervening more quickly instead of waiting a long lifetime for
Yeah,
interventional impact.
so I'm really interested in, I'm trying to figure out how to study the wildfire issue, which is, I keep thinking like, oh, I missed the next one. And then the next one comes and, it's going to be fires in Canada forever. The other thing is my colleagues, Ushi Graham is studying military survivors of burn pits. So they were burning, terrible things in the Gulf, in the Middle East. And so we're hoping to look at that as a good question too. And I think there's also been some nice work of 9/11 survivors in New York. So yeah, I think we really have to think carefully about what's the right population to kind of squeeze that timeline so we can really see the effects.
Excellent. Thank you again for a phenomenal presentation.
Thanks so much. Thanks for having me. It's been a wonderful discussion.
Thank you all. Have a great night.